you have seen now 3 very different approaches. You will see 2 more that are very different. First, we saw now a separate platform, programmed, created by his father, whom you met, Bruno Meissonier. Then you saw Professor Paulo Dario's approach, which studies nature, mimics it, finds the mechanical machines in it and the robotic movements, and how from that we may in fact go back to create new science, not just in neuroscience, but also for biomedical science. Now you've seen Professor Serageldin talking about not just creation of androids, humanoids, and geminoids, but the kind of interaction and proximity that begins to occur between the appearance of a human being and the appearance of a machine, and whether there is convergence between them. We'll take a brief break and we'll come back for the next session. We'll have 2 great pioneers who have taken us further, I think. Miguel Nicolelis will tell us about the links between reading what the human brain is thinking and having machines implement it. And Kevin Warwick rightly can claim that he is the first human being on Earth to be a cyborg. I've asked my colleagues, for those who have questions, to put them on paper so that we will assemble the questions for the previous session and for this session at the same time. The questions will then be passed on to Dr. Rafiq Nakhla and Dr. Muhammad Faham, who will assemble the questions, regroup them, and give them to us. And we will have all 5 speakers and myself in a debate in the last part of this session. So we have 2 speakers in this session, followed by a discussion group where all 5 speakers will join me on the podium. So our next guest is really a very remarkable scientist, one whom I have admired for a very long time. I've tried to get him to come to Alexandria for a long time, right, Miguel? And I'm happy to say, things didn't work out, but they worked out beautifully for him to be here. Professor Miguel Nicolelis is professor of neurobiology, biomedical engineering, and psychological and brain sciences, and co-director of the Center for Neuroengineering at the Duke University Medical Center in the United States. But he's also from Brazil, and is also a wonderful leader in transforming Brazilian scientific research, and he created the whole program of scientific cities, but that is another story for another day. Today we will stay with the neuropsychological work that he's doing. He is the Deane Professor of Neuroscience and Professor of Neurobiology, as I mentioned. Now, he is a native of Sao Paulo in Brazil, and he has really been best known for his pioneering studies of brain-machine interfaces. So if you hear the letters BMI coming up here, it is not BMW, it is BMI. And the intention of BMI, when it's in connection with Miguel Nicolelis, is brain-machine interfaces. And he's looked at neuroprosthetics in human patients and non-human primates. Now, what he did, really, he developed an integrative approach to study neurological and psychiatric disorders, including Parkinson's disease, epilepsy, schizophrenia, and attention deficit disorder. And he made really fundamental contributions to the fields of sensory plasticity and reward and learning mechanisms and feedback loops. But what makes it very powerful, I first got to know him based on his pioneering work of being able to read the brain waves of a beautiful little monkey called Belle, and ship these brain waves the thoughts of the monkey by wireless internet 1,000 kilometers away, 1,000 kilometers between Duke and Massachusetts, and a robotic arm would move based on the commands of the brain waves of the monkey. Subsequently, he went much further, and with colleagues in Japan, he did fantastic experiments that read the brain waves of a monkey in the United States, affected a robot in Japan, then got the sensory data from the robot in Japan that then affected the behavior of the monkey in the United States. And I'm sure that since that last experiment was 2008, so I'm sure he has a lot more exciting things to tell us. Ladies and gentlemen, please join me in welcoming the one and only Miguel Nicolelis.
Thank you very much for this kind introduction. It's a great honor, a privilege, and a great pleasure to be here today, here in the home, the first home of knowledge, the first place where humanity recognized that it was important to collect everything that we produce from our brains for future generations. So it's a true privilege to be able to speak in this podium, in this place. Well, I'm going to change a little bit gears here. I'm a neuroscientist, and we heard a few lectures, during these few lectures, a lot about what is to be human. In fact, I want to talk to you about that in a different degree, from a different point of view, and showing to you that what I have been doing for the last 25 years, reading electrical brain activity from a variety of species, including humans, is actually a search for precisely that, what is to be human. If you've never seen a true brainstorm, this is 10 seconds of one. These are 10 seconds of about 100 neurons of a primate firing. Each little vertical bar that you see in each line of this matrix is a single brain cell firing electrical sparks, all together producing something that we know colloquially as a thought, a motor thought. And my job has been to try to look at patterns like this, thoughts like this, and extract from this seemingly apparently random sparking of neuronal activity and extract meaning out of it, and try to prove that you can not only read these brainstorms, but you can actually decode the meaning embedded in them and use this meaning, the voluntary will embedded in that raw electricity, and use it to reproduce the desire of that subject into a machine that is going to be under control of that particular brain. So what I'm going to show to you is, as you heard in the kind introduction, is how you can connect brains and machines in a closed loop control, and how you can actually endow machines with a brain and allow human and animal reach to be enlarged by the qualities that a machine gives to the human body. If I get to the end of this, in the time allocated to me, I'm going to show you that when you do that, you get to a point that we have just reached, where the brain is fully liberated from the constraints and limits that the body imposes on it. That you can actually establish a bidirectional linkage between living brain tissue and a device in which activity from the brain tissue controls the movements of that device, and feedback information from that device can be delivered directly to the brain without the interference of any part of the body. So let me start by showing to you what a brain-machine interface is. This is the original graph that we published when we defined this term. And what you see is a brain, a human brain, but it can be any brain. And these are called electrodes. These are hundreds of metal filaments that can be chronically implanted in the brain so that we can harvest the raw electrical activity today of about 500 brain cells simultaneously. And then using a variety of digital and electronics, microelectronics technologies, decode the information embedded in these signals and transmit these voluntary motor commands that are embedded in this raw electrical activity to machines that can actually enact the voluntary will of the subject. Well, we can then close this control loop by sending feedback information, sensory-like information from these devices back to the brain. Until today, using either the visual system, so that the animal looks at images or the machine itself, or the skin as a tactile transducer. But just recently, we achieved the goal that we pursue for almost a decade. That was to actually link this machine directly with the brain, using the same type of electrodes to deliver sensory information in the format of electrical microstimulation, up to the point in which the animals learn to decode that information and close this loop without the need of using the body. Why we're doing that? We're doing that first to test a series of principles of how large populations of brain cells that define those brainstormings, that define actually the essence of what we are, because embedded in patterns like what I showed to you in the first slide is pretty much everything that every one of us has ever done throughout the history of mankind. Everything that we do, remember, plan, feel, regret, dream, or endow, give, comes from brainstorms like that. That's the essence of what a human being is. But there are moments in the lives of some of us in which, because of trauma or disease, parts of this wonderful system that is the nervous system may be damaged, like for instance in the cervical spinal cord lesion. And then our desire to move, to roam freely through the world is interrupted. Even though we can continue to imagine and dream about moving, we can no longer enact these wills, these thoughts, because this pathway that carries our dreams of moving is interrupted. So these signals cannot reach our body. We are paralyzed. The idea is to use these brain-machine interfaces one day to actually bypass these lesions, harvest the signals that are still produced by the brain, bypass the lesion with microelectronics and robotics, so that a patient can imagine again how to move, and move again, because now he's going to be wearing a new body, a wearable robot, an exoskeleton that is going to be controlled by his own brain, and it will allow him to once again move freely around the world. That's the second reason we are doing this work. This is 20 years of different electrodes, 3 generations of microelectrodes that can be implanted in the brain to harvest brain signals. We have developed all this in our lab, but we have realized that there was an innovation that we are capable of doing. It took us several years to get to this final design, where we can implant hundreds of these electrodes, close to 700 to 800 electrodes. But instead of only recording the electrical activity at the very tip of each one of these electrodes, now we have active sites throughout the shaft of each one of these electrodes. Something like a tree. Each one of these contacts, and we can have 15 to 20 per shaft, can record the electrical activity produced by an individual neuron. So if you have a 10 by 10 and 15, you already have 1,500 contacts per probe. Since you can implant 10 to 20 of these probes per monkey, you can get to thousands of active contacts. And what we are about to do is to jump from 500 to 50,000 individual neurons recorded simultaneously. We have just a little sample of how that may look like. I'm going to show you very briefly. It's our first prototype that worked. And you see 4 of these cubes, we call them cubes, sampling simultaneously through the X, Y, and Z, the depth of cortical areas. This move is 100 times slower, so every time you see a flash here, a neuron has sparked, has fired an action potential. So we got about 500 cells recorded simultaneously in 4 cortical areas while a monkey was moving around. This is a brainstorm. This is a symphony. This is a neuronal symphony. This is actually a thought. And if this was not a monkey, if this was a person, a human being, this would contain a piece of the essence of what we are. Well, let me show you what we can do when we record the activity like I showed to you. Oh, you can pick it up, that phone. It must be mother. This is what you just saw. Each one of those little balls firing was producing an electrical action potential. If you look at all of them simultaneously, you have what we call a neuronal ensemble. This is a term, a jargon, but defines a population of neurons firing together. So what we did was to create this type of paradigm in which monkeys are trained first to play video games with a joystick. And they use this joystick to play the game while we are recording hundreds of neurons simultaneously and running this neuronal activity in real time to a series of computers. They are running models that actually try to simulate what a spinal cord does in the real human being. And this poor man's spinal cord is trying to extract from the raw brain activity the type of commands that this monkey uses to move a joystick to play the video game, so that it can reproduce, now in digital format, in a robotic device, exactly the same movements that the animal is making to play the game. This is the first part of a brain-machine interface. The second is to send feedback to the monitor so that the monkey can see how well he's controlling this device by thinking about the movements. Because in a given part of the experiment, we play a dirty trick with our monkeys. We actually remove the joystick, we turn on the brain-machine interface, and now, for the monkey to get the reward that he wants to get every time he plays a game and gets it right, he has to simply imagine the movements, because his arm now does not have an actuator to play the game. The cursor, the computer cursor in the screen is now controlled by the robotic hand of this robotic device that is now under total control of the animal's brain. So the only way for the animal to win the juice, the orange juice, that he craves as a reward is to actually imagine how to play the game and control the robotic device to actually do it. I'll show you that this can be accomplished now also by sending feedback later on, I'll show you, directly to the brain. But first, let me show you the simplest case. So this is how this game works. We record all the neuronal activity. We send to a series of simple linear models. They are looking back in the past to predict the future of these movements. And we send these parameters now in digital format to simple robots that are going to perform the job that the monkey needs to perform. So this is one of my favorite monkeys, and her name is Aurora. And as you can see, she loves to play this game. And the game is very simple. A big circle appears on the screen. Aurora has to move the joystick very quickly and cross the center of the circle. Every time she does that correctly, she gets a drop of Brazilian orange juice. And for Brazilian orange juice, you do anything in your life, I can guarantee you. Our monkeys agree with me, I can prove. So you see that she's learning to play the game, and while she's doing that, we are recording her brain activity, sending to computers, translating her raw electrical brain activity into digital commands, and sending to a robotic arm that is going to play the game for her in a few minutes. And the reason we know that is that just by recording between 100 to 300 neurons simultaneously, we discovered that our simple linear models, our poorest man's spinal cord, could predict up to 21 degrees of freedom simultaneously coming out of the cortex. We could actually predict kinematics and dynamics, muscle patterns of activation, onset of movement, all sorts of parameters that are necessary to replicate the movements of that primate arm into a robotic device. And that is the moment when we did the trick. We removed the joystick from Aurora, we turned on the brain-machine interface, and to our total surprise, she just got it. Every trajectory that you see now produced by the cursor to reach the target is derived in real time from the raw brain activity of Aurora's brain. And as you can see, she doesn't need to move anymore. There's no point in activating the muscles. In fact, if I would show you the muscle patterns of all her body, you would see nothing. They all relax because she realized that she only needs to imagine the movements. The brain-machine interface will do the job for her. It's the dream of every Brazilian. You just imagine, and someone else does the work. In this case, someone else is a robotic arm. That was in a different room at Duke University, but it could be anywhere. As you can see, it could be in Japan. It could be on the surface of the moon. It could be in a different planet. The arm is now playing the game, and 30 days after learning this trick, Aurora realized that she can use her biological arms for something else. She can scratch her back, she can scratch one of us, she can pick up food with the other hand. By virtue of the process of plasticity, cortical plasticity, the robotic arm is assimilated into the body representation of Aurora in her brain. In fact, this monkey now has 3 arms, 2 biological and 1 mechanical. And she can control all 3 with her single brain. Well, is this only true for the upper limbs? Can this be done for other parts of the body? The answer is yes. This is a rhesus monkey that was trained to walk in a treadmill, like we do, like we exercise. And as you can see, the joints of this monkey are painted with fluorescent materials because we have cameras all over the room taping the kinematics of his movements. And we are doing the same thing. We are sending hundreds of brain cells activity to a series of computers and trying to reproduce the bipedal locomotion patterns of these monkeys in a treadmill. This is the raw brain activity that when you combine from different parts of the cortex, the sensory and the motor cortex, what you can see in the predictions of our models in blue is that we can really reproduce very well the step cycle of this monkey in real time using just raw activity from the cortex. But here, we can reproduce the step movement at different speeds in random parameters forward and backwards. We can actually generalize for a variety of movements in the treadmill. At this point, with our good friends at ATR Kyoto, Mitsuko Wato and Gordon Cheng, we decided to play an even bigger trick with our monkeys. I guess we can call this the first transcontinental brain-machine interface test. Actually, what we had was a monkey at Duke University walking a treadmill. We are recording about 300 brain cells simultaneously, and we are sending this activity to Kyoto, to ATR laboratory, where a humanoid robot, CB1, actually performed the dream of his life to walk like a primate. Because using these brain signals decoded into digital commands, we could get CB1 to walk. And using cameras around, we could send visual feedback back to Duke and project the legs of CB1 walking in front of the monkey so that the monkey could actually have the impression that those were its legs walking. So we actually closed this brain-machine interface, and it worked on 220 milliseconds to send the signals to Japan and get the feedback back. About 20 milliseconds faster than it takes for an action potential from the brain of the monkey to get to the monkey's leg. And what we actually did, and this is a more recent experiment, is to get CB1 walking using brain activity from a primate recorded across the globe. Sure, it's not Hollywood type of walking, but it's walking nonetheless. The interesting thing is that when we actually quantify how many neurons we needed to actually get very good signal-to-noise ratio and produce walking in different ways, each one of these colors represent a different brain area, and the black dots represent all the areas that we recorded simultaneously. The big surprise we got is that with about 150 to 200 neurons, we could actually get that robot walking, or those upper limb robots moving. And the signal that we needed was widely distributed all over the cortex. This message was not in a single place, was not in a single cortical field, not in a single homunculus, as we call. It was all over, widely distributed. So we could actually sample, and if we combine these structures together, we got a better output to actually control our robots. Well, but that, if you want to get to a prosthetic device for a human being, that's just half of the story. Get motor signals out of the brain and make a robotic device actually walk, move the arms, and we have just demonstrated recently that control balance can also be done with cortical signals. We can control the balance of the robot with cortical signals. But there's the other part, the feedback. That's when we design these experiments, and we call this a brain-machine-brain interface, because now, not only the animal has to control a device and move the device in a virtual environment, that device sends feedback directly to the brain to inform the animal that that's the right type of movement, or that's the right type of object that the animal has to find in space. And this is done by sending codes, electrical messages, back to the brain, and by training the animal to recognize this message as if these messages were delivered to the skin of this animal. Actually, I'm going to show just one task, because it's perhaps the most important one. It's very similar to the first movies I showed, but in this case, the monkey has to use its brain to put a cursor in the center, and then two targets appear. Visually, these two targets are absolutely identical, so the animal cannot know visually where to move the cursor to get a reward. The only way he can discover is that as he moves closer to the correct target, he's going to get a message in the brain that says, this is the right one, grab it. If he moves to the wrong target, there'll be a different message saying, no, no, this is the wrong one, or there'll be no message. So the animal can only identify the correct virtual object to grab with its brain activity alone by interpreting the message that is sent back to the brain in this bidirectional dialogue. You are going to see in the movies, there is a gray zone indicating what is the correct target, and there is a little green arrow that shows what is the correct target, but the monkey does not have this information. This information is for you. The monkey has to do it just by sensing with its brain the right message. So we first did this testing with tactile feedback using the skin. And in about 14 sessions, the monkeys got close to 90%, 95% correct. When we used electricity instead of tactile message and delivered directly to the brain, it took longer. It took a month. But the brain got it. The monkeys got it. And that's how it looks like. So this is when the monkey is being trained. Is using the joystick first to move the cursor, but is learning the message, how to interpret the correct message. So he first centers in the center, waiting for the two targets. He got lucky in this time, but now he's going to get it wrong in the first attempt. Immediately he moves there, and when he gets in that gray zone, he gets a buzz in his head that tells, this is the right one to do it. And you cannot hear because the sound was not connected, but there is a sound here that I'm using to illustrate to you that this is the correct target. So the monkey is learning very quickly, and after about 30 days, we do the trick. We remove the joystick, and now everything that you're going to see in the next video is done by the brain. The brain is moving the cursor, centering the cursor, is probing the two locations to find the correct one, and when he finds the correct one, it stops at the center so the animal can get the reward. That is the moment in which the primate, the brain of that primate, was fully liberated from its body, because it could execute this behavior without the need of any receptor in the skin or in the eye, any part of the body, any effector. It could do it exclusively by linking its brain to a computer device. And that's it. Again, it's centering, finding the right target, getting the reward. It centers again, looks for the right target, gets it, gets the reward. This is after 30 days. You went to the wrong one, didn't get it, find the right one, get it. You can take pictures, by the way, of everything. It's all free. You're witnessing, you may perhaps not notice, a brain that doesn't need a body. Of course, it needs a body to survive. It needs a body to get oxygen, to get the metabolites, but it doesn't need a body to act upon the world. And this was the first time that it ever happened. Why we want to do all this? Well, because we have this thing called the Walk Again Project. We are building a non-profit international consortium for the next 5 years to make someone walk again. A young Brazilian kid or a young Egyptian kid that will be able to use his or her brain again to control a body. It's going to be a different body. It's going to be a robotic body, but nonetheless a body that will give freedom again to that brain that it has become a prisoner of a body that cannot correspond to its will and its desires. We already have a prototype of this body. It's going to be an exoskeleton, a wearable robot, that this person, as you can see, has the right football colors too. These are Brazilian colors for the Americans in the audience. I'm from there, I know, they don't know. So the idea is that all that I showed to you is going to be used to control multiple degrees of freedom of this exoskeleton that Gordon Chang is building at the Technical University of Munich, and the different sensors all over this exoskeleton are going to send messages back to that brain wirelessly, so that that brain can be assimilated as part of the self of that subject, part of the brain map of that body, because I heard it before in one of the talks. I think I got it wrong, but I'm going to say what I think. We only think that we have a body because our brain simulates the existence of our body. Our body could be anything. It can be anything, provided that the brain wants it and gets some advantage of doing it. The brain is the greatest simulator we ever seen this side of the Milky Way, as far as we can prove. And as a modeler, a simulator, it can create reality and a representation of self the way it wants. So our prediction is that these patients are going to basically incorporate this exoskeleton as their real body, and their real body is going to roam free through the world under the control of their brains. Well, we are doing something preliminary to test this concept. We built a monkey avatar, and I have to say that this was way before the movie Avatar. So I'm looking for Mr. Cameron from some royalties, because we actually had the experiments before he came up with the movie. This is a true realistic kinematic avatar that is interacting with monkeys, because we want to prove the concept first in monkeys that a whole implant we already have for the entire motor cortex. The entire body representation can be used to control the movements of this avatar in a virtual reality environment. And we are already running these experiments, and the moment we conclude that, we are ready to go to human trials. I have to tell you that it's impressive to see how the monkeys think that the avatar is one of them. All the behavior signs when they interact with polarized goggles with this avatar that looks three-dimensional to them and moves around according to their movements or their brain activity, they actually think that they're in the middle of some colony You can put one or two or many. And they use the kind of mouth snapping and the kind of vocalization that they use when they're interacting with their kin. So they recognize the avatar as a real guy. I decided to bring just a simple animation since we're approaching a very, very important event this summer. We have a student from Cambridge programming the avatar for our monkeys, and one student from Lausanne, Switzerland. And as you can see, the avatar can do very spectacular things, like kicking a penalty kick as a guy from Cambridge. It's not a Brazilian style, but he would do it. So this kind of detail can be programmed in these avatars. And I hope we come here one day soon to show you the whole avatar being controlled by the monkey's brain activity alone, no movement necessary. Well, I'll just finish by saying that once you tap into this, once you open this window, once you try to liberate the brain from the body and you start exchanging messages with living brain tissues, funny things can happen that you never expected would happen. Where at the same time that we are doing these experiments with monkeys, we are working with mice, transgenic mice. They are very good models of Parkinson's disease. And as you can see, these are two movies, by the way. These animals are completely frozen, completely akinetic. They are terminal Parkinsonian animals. They cannot move at all because they have been depleted of dopamine completely. So they are frozen, like the movie, a much better movie than in which the patients were completely frozen. Oliver Sacks, I think, wrote that book. Well, what we realized by recording many, many neurons simultaneously is that Parkinson's disease is nothing but a particular type of epilepsy, a particular type of seizure, where neurons start firing all at the same time in particular motor areas of the brain. So if you treat the animal, this is the seizure, with the typical medication for Parkinson, the seizure goes away and the animal moves. Well, there is a procedure, a surgical procedure that is very invasive called deep brain stimulation, where you put probes 10 centimeters deep into the brain and you start stimulating terminal Parkinsonian patients, and you get very good results in terms of allowing them to move again. But it's very invasive and only 5% of the patients can take it. Well, when we developed our techniques to stimulate the brain, and we developed our new principles or theories of how the brain processes information in a distributed way, we try a very different approach. We try to basically stimulate the surface of the spinal cord. We didn't even open the tissue. We just put on the surface of the spinal cord stimulating electrodes and use high-frequency stimulation like we use in the experiments I showed you with the monkeys to stimulate the entire motor system and somatosensorial system of these animals that were terminal Parkinsonian animals. What we found is that the moment we stimulate the surface of the spinal cord, and I'm very sure that this will be done non-invasively, completely from outside, we disrupt the seizures and the animals move again, immediately. No need for medication. This is one of these guys. It is 80% depleted of dopamine, so it's a very severe Parkinsonian mouse. He's shaking and he cannot move even though he wants to drink the water here. But as you can see, he is frozen. It's frozen completely. All muscles are contracting at the same time. He cannot move. At 5:50, we are going to start the high-frequency stimulation of the spinal cord in this animal. 2 seconds, and you're going to see the effect. It's instantaneous. The seizure is disrupted. The neurons are firing off phase now, and the animal can move like he used to move when it was normal, can go drink the water and do everything that a normal mouse needs to do. Here now, the stimulation is going to go off, and you're going to see that in about 10 seconds, the symptoms of Parkinson return and the animal freezes again. But as long as we keep that stimulation going, the animal is going to move. It's going to continue to move. This procedure would cost a tiny little fraction of deep brain stimulation, every patient will be eligible for it, and you could start the minute you're diagnosed with Parkinson's disease. You don't need to wait until you get severely sick. So this is how we are going. This is what is going to happen in the future of this field. We thought that the brain was a place that we could not touch, that we could not enter, that its secrets were too holy, too difficult to break, almost impossible to establish a dialogue. That is all in the past. The door is open, the dialogue has started, the future will be bright. The conclusions here are exactly what I told you, so as a good Brazilian, I'm not going to repeat them. But what I want to tell you is, the future will be bright, but in my opinion, there will always be something that is human that we cannot transfer to a machine. Because besides the brainstorms and the electrical activity and the circuits, we have a unique evolutionary and individual history. We have, like this place, millions of years of history behind our brains, and that nobody can simulate. Thank you.